Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, with our ongoing series on Introduction to Proteins, today we are conducting another session. Today we will be talking on protein degradation and recycle, and specifically we would be talking on lysosomal pathway. And for this discussion we have once again with us in our studios Dr. Unnati Gulati. Dr. Unnati Gulati is academic coordinator in CEC, and we would like to share with you all that she has taught in numerous colleges in the past, as well as she has written or I would say she has contributed in various research papers. So we would like to welcome our guest Dr. Unnati Gulati, and let's try to grab maximum knowledge through her. Hello ma'am, welcome to the research lecture.
Thank you Geetika. Dear students, welcome to this lecture. In the previous lecture we were talking about protein degradation through the ubiquitin proteasome pathway, and we had said that today we will be talking about lysosomal pathway. Many of our thoughts are that lysosomal pathway will be very simple and there is not much to learn about it, but as we go through this lecture we will come across many fascinating facts and in fact many fascinating discoveries that have occurred with the study of lysosomal pathway. So continuing with that, we have been talking about misfolding and misassembly of protein as the starting point for where we have to look into degradation of proteins which cannot be taken care by either the chaperones or other machineries. So when we have the on pathway folding going on, the protein will reach its native state and will give a proper appropriate function. But when there are chances of an off folding pathway occurring, then there are some accidents in folding and the protein may become misfolded, non-functional, or may form aggregates, and aggregates can be of several types as we have discussed in several diseases, the fibrils, the plaques, etc. So what happens when the protein has formed these irreversible products? So what is the next step to reclaim them back? So we had talked about ubiquitin pathway taking care of about 80 to 90% of such accidents, and what happens to the rest of the 10 to 20% of the proteins generally which are extracellular, also the cell organelles, and some of the intracellular proteins also may fall under the lysosomal pathway. So we agree that the cell is constantly in a dynamic state of synthesis and degradation. There is a constant recycle of nutrients and building blocks. To eliminate abnormal and excess proteins, there are various pathways, and there is a regulation of the metabolism and regulation of each and every step in degradation as well as recycle. So again, to summarize what is protein misfolding, we can have stochastic fluctuations, we can have destabilizing mutations, excessive oxidative damage, we can have aging or cancer related changes or mutations in the protein. Some stress may be environmental or physiological. There can be certain post-translational modifications which may result in the protein becoming non-functional and misfolded and may accumulate to give deleterious mutations and accumulate to give several plaques which burden the cell and cause aging and result in several diseases. So what is the chemical stability which gets disrupted can result in misfolding or degradation or disruption of the structure of the protein. So firstly we will talk about the reactive oxygen species. As you can see, some of them are depicted here. These are the unpaired electron containing chemical moieties: oxygen, superoxide anion, peroxide, hydroxyl radical and hydroxyl ion. See, all of them are showing an unpaired electron, and this gives them a very high reactivity, and they can in fact damage your lipids, sugars, DNA, proteins, and form such adducts such as the 4-hydroxy-2-neonal, and this is a marker which can be studied and which can be sought and seen in several cells which are undergoing cancer or which are showing aging. So it is a marker for oxidative stress. Several other chemical reactions can occur in which there is deamidation, isomerization and racemirization of the aspartyl groups, which can result in the protein having a changed structure and may result in loss of its function. So, we have to reclaim back the building blocks. So, what we can do? We can break down the chunks of protein which are non-functional and reuse the amino acids to form new and new proteins. Similarly, that will apply even to your DNA or lipids and RNA too. So, this is the basic of protein peptide bond hydrolysis, and this is the reaction which will result in breakdown of the long chain peptides, polypeptides in the protein to their constituent amino acids. So, here we are seeing either you can use heat and hydrolysis in presence of acid, or heat will result in breakdown into the respective amino acid groups, amino acids residues. So, protein homeostasis is maintained by the cell by this degradation as well as synthesis pathway, and there are several checkpoints where the quality of the protein existing in the cell is regulated. So, this can be regulated either by the chaperones that are existing, or it can be regulated by the endoplasmic reticulum, and it can be then taken care of by the ubiquitin pathway or the lysosomal pathway. So, there exists this protein quality control system, and it is having several components. So, when we have talked about protein hydrolysis, which was occurring definitely in a chemical reaction. So, what is the chemical reaction? How is it assisted within an organism? So, what are the enzymes or catalytic moieties which are responsible for such breakdown of protein? So, simply we will start with several type of proteases or those enzymes which can break down protein. So, the first type we are going to talk about is the serine protease. Most of your and all of your digestive enzymes, the trypsin, chymotrypsin and elastase are of this type, and they have serine and histidine and aspartate in the active site. So, during the nucleophilic attack, so a proton will be transferred from the serine hydroxyl oxygen to the imidazole group of the histidine, and this is going to be just adjacent to the aspartate carboxyl. So, this 3 amino acids are very important for carrying out proteolysis in serine proteases. Simultaneously, another protease will be aspartate protease, and another digestive enzyme pepsin, some other which are found in lysosomes, and we will be talking about them. Renin and HIV protease contain aspartate or 2 aspartates within their active site, and in this one it shows acid base type of catalysis, in which one aspartate will accept a proton from the water and will attack the carbonyl carbon of the peptide linkage, and the second aspartate will donate a proton to the oxygen of the peptide carbonyl group, and this way it will cause hydrolysis. Another type of carboxypeptidase or metalloprotease is the zinc containing proteases, and several matrix metalloproteases such as collagenases are secreted by cells. Some of them are lysosomal, and some are involved in degradation of extracellular matrix during tissue remodeling. Some have role in cell signaling, and some are able to release cytokines or growth factors. So, they assist in signaling and regulation, and they are characterized by having a zinc binding motif in their active site, and 2 histidines are going to tether to the zinc. So, this zinc promotes the nucleophilic attack on the carbonyl carbon of the oxygen in the water, and it is part of the active site. Another type of protease would be the cysteine protease, definitely containing cysteine sulfhydryl group as the active site. Example of this are papain, and this is found in papaya. And then we have cathepsins, which are found in lysosomes, and caspases also found in lysosomes, and calpains. So, caspases are involved in activation and implementation of programmed cell death, and they cleave on the carboxyl site of the aspartate residue. Calpains are calcium dependent, and they are activated by calcium, and they cleave intracellular proteins involved in cell motility and adhesion. They regulate process such as cell migration and wound healing, and also result in the meat becoming tender when we marinate it. So, it is used in meat tenderization. So, calpains are of two types, calpain 1 which requires calcium in millimolar quantities, while calpain 2 requires only micromolar quantities of calcium for its activation. And these proteases have domains comprising of two domains that fuse to form a functional protease only in the presence of calcium. And they show proteolytic processing. Unlike ubiquitin, which is only a tagging mechanism and it passes on to the proteasome, calpains are tagging as well as they show proteolytic mechanism or in proteolytic processive mechanism, and they are involved in substrate recognition and they identify certain short amino acid sequences on the proteins that need to be degraded. So, coming on to the organelle that we are going to focus on is the lysosome, and lysosome is a membrane bound intracellular compartment having several about 50 such hydrolytic enzymes, the hydrolytic enzymes examples of which we have just discussed. So, this lysosome is the double membrane bound intracellular organelle, and it is characteristic in having a slightly acidic pH of about 4.8 to 5.5. And how does it maintain this internal acidic pH? Even though in the cytosol you will have the pH of around 7.2. So, this is very essential component because all the lysosomal enzymes that we are going to talk about are active only at a low pH of 4.8 to 5.5, and this lysosomal enzymes are inactive in case there is a rupture or leaking of the lysosomal enzyme into the cytosol, just because the pH has now become more neutral and they are having an optimal activity at only acidic pH. So, lysosome are found in the cell and they are thought to be generated from the maybe either from the plasma membrane or from the Golgi body, and they are bud from the compartments of the late Golgi. They are taking part in the trafficking and sorting, and they bind those or engulf those proteins that need to be degraded, and there is some sort of signaling selectivity also encountered in this kind of degradation. And lysosomes not only take care of proteins which are present in the cytosol, but they also degrade those proteins which are found in the membrane, and hence lysosome also contains several lipases along with the protease to completely degrade the protein and the membrane. So, as you can see in this diagram also that there is ATPase which is creating low internal pH. So, these ATPase are called vacuolar ATPases. Lysozymes are organelles which will be encountered in larger organisms in mammals. When we are talking about unicellular like yeast, we will call these organelles as vacuoles, and vacuoles have specific ATPases. These are the type of AAA ATPases, and these are ubiquitous, they are multi-subunit proteins much like your ATPases which you encounter in the mitochondria, and this is located in the acidic compartment. They are endosomal lysosome and they are vacuoles in yeast. So, in vacuoles you will find the ATPases. A constant ATPase can generate enough flux to get all the protons into the vacuole and create an acidic pH, but after some time there is going to be a disbalance of ions and hence no more acidification can occur. So, along with this you will encounter a chloride channel, and many research papers will tell you that CFTR like chloride channels may be present on lysosome. These channels are also responsible for lysosome fusion, and they balance the positive charge by bringing in the chloride, and hence acidification can occur and pHs of 4.8 to 5 can be reached. So, these ATPases occur in conjugation with chloride channel proteins. So, where does the lysosome come about from? These lysosomes are very transitory organelles, and they are formed from phagocytosis, endocytosis and autophagy. So, these may be formed from the Golgi complex or from the rough ER, and the Golgi complex sorts the lysosomal enzyme in the trans region, and it attaches the phosphate to the mannose residue, forming the mannose 6 phosphate, and this forms a sorting signal. Now, it moves through the cisternae to a trans region and binds to a specific receptor. A bud is from the Golgi complex, and a cage is formed outside, which is made up of clathrin, and this clathrin enclosed vacuole now further moves to a lysosome. So, there are, as we were saying, 50 such hydrolytic hydrolases, acid hydrolases, which may degrade proteins, DNA, polysaccharides and lipids, and they are optimally active only at a acidic pH, and they are inactive at neutral pH of the cytosol. Much so, the mechanism to prevent accidental autophagy or accidental breakdown of the cell in case of rupture of a lysosomal membrane. And lysosomal enzymes are secreted like proteins which are destined for secretion or which are going to be resided in the plasma membrane, but because of the recognizing signal, they are recognized by the phosphotransferase enzyme as soon as it is leaving the endoplasmic reticulum. So, this phosphotransferase transfers the N-acetyl glucosamine 1 phosphate to one of the mannose residues, and the glucosaminidases next removes the glucosamine to generate the M6P, which is the mannose 6 phosphate. Acid hydrolysis are targeted to lysosome by mannose 6 phosphate residues, and they are recognized by the specific mannose 6 phosphate receptors in the trans Golgi network, and they are packaged, as we were discussing, into clathrin coated vesicles which fused further with the endosome and finally forming a lysosome with acid hydrolysis. Once the fusion has to occur, the clathrin coat is removed and the transport vesicle fuses with the late endosome. This late endosome eventually will form the lysosome, and the acidic internal pH causes the hydrolases to dissociate from the mannose 6 P tag P receptor and release the enzyme into the lumen of the endosome, while the receptor will remain outside in the membrane and will be recycled to the Golgi. So, this way the lysosomal enzymes travel from the ER to the endosome and are fused with it to remain active within the acidic compartment of lysosome. Some of this is the extensive list of acid hydrolases found within the lysosome. So, you can see there are glycosidases, there are lysosomal proteases and sulfatases, and the ones that I have highlighted. So, there are 50 such enzymes and 50 such diseases or syndromes can be encountered with the mutation or absence or deficiency of any of these enzymes. So, malfunction of the lysosomal enzymes can result in diseases, and 50 such have been reported. So, Gaucher's disease is a deficiency of glucocerebrosidases, and Christian Dedoux, and this name will come across again, in the year 1960 suggested that exogenously if you administer such enzymes, they might be taken up by endocytosis by the lysosome and the deficiency can be met with, and the enzyme can be made functional. So, this enzyme glucocerebrosidase breaks down the glucocerebroside into a glucose and a cerebroside, and hence accumulation of this glucocerebroside causes the Gaucher's disease. Another disease which is due to the deficiency of hexosaminidase A, which is Tay-Sachs disease, in which the gangliocytes accumulate in the brain nerve cells. This will result in neurodegeneration. And the third one is the acid alpha glucosidase enzyme, which results in the Pompe's disease. In this, accumulation of glycogen occurs, and this leads to progressive myopathy or muscle weakness, and it may affect the heart, skeletal muscle, liver, and also the nervous system. So, we were talking about ubiquitin and previously. So, now let us look at briefly what lysosomal proteolysis has. It requires basically lipases and proteases. Also, it may require some lysosomal chaperones which may occur on the membrane of the lysosome. So, the lysosomal membrane receptor LAMP2A provides a pathway across the membrane, and several proteins which have been brought in by the chaperone can be taken up by this lysosomal membrane receptor. So, there are chaperones which assist in the translocation, and lysosomal proteases which also include cathepsin, aspartate proteases and zinc proteases, apart from this among the list of the 50 that we discussed about, bring about the degradation of the engulfed protein or the cell organelle or other components. So, there are two major functions which we encounter in lysosomes. The first one is endocytosis. Endocytosis is also the basis of formation of lysosome if it is being formed from the plasma membrane, and autophagy is another component which we will discuss in details. So, endocytosis is an active transport of proteins into the cell by engulfing them in an energy driven process. Here you will see transport of very large molecules that cannot pass through the hydrophobic plasma membrane or through the channels. In this, endocytosis term was also given by Christian de Duve, and it has 4 types: clathrin mediated endocytosis, claviole mediated, micropinocytosis, and phagocytosis. So, phagocytosis, which occurs in macrophages, can take up particles as large as 0.75 micrometer in diameter, and this may include your bacteria, cell debris, aged cells which are in the phagocytic vacuoles or the phagosomes, and these, when fused with the lysosome, they form the phagolysosomes. And discovery of endocytosis is by Elie Mechkinoff, and so we will talk about ER quality control. Endoplasmic reticulum associated degradation of proteins. Many a times a protein which is formed from the ribosomes attached to the endoplasmic reticulum or the rough endoplasmic reticulum, it releases the polypeptide into the ER where it eventually folds. In case there is any misfolding, there are several chaperones existing in the ER which will take care and cause or assist its folding to appropriate folding, but many a times any polypeptide which skips or is unable to form its appropriate structure, it is designated for destruction, it is marked and designated for destruction, and this protein is translocated back to the cytosol, and in the cytosol you have all the ubiquitination machinery occurring, and this protein gets ubiquitinated and degraded by the 26S proteasome. So, autophagy, if you remember from your class 11-12 books, we used to learn the terms autophagy and who coined it, but we never realized all the implications and all the mechanisms that underlie such a simple looking process. So, in the year 1974, Christian de Duve, Albert Claude and George Paladay received the Nobel Prize for their discovery, which occurred in the year 1950, where they discovered lysosomes. And they found that, and they also studied autophagy, and by the year 1963 they coined the term autophagy. So, they found that in well nourished cells, lysosomal protein degradation is non-selective. In starving cells, however, this degradation would deplete essential enzymes and regulatory proteins. So, early 1950s, a Belgian scientist, Christian de Duve, studied the action of insulin, and while he was studying that, he found he wanted to look at the intercellular localization of glucose 6 phosphatase, and they were doing cell fractionation. Cell fractionation is a technique in which we break down a tissue and we break down the plasma membrane, and using centrifugation are able to fractionate or separate all the organelles based on their size or sedimentation coefficient. So, they were trying to localize glucose 6 phosphatase, and they encountered on the distribution of acid phosphatase. So, when this fresh liver extract was found, there was no enzyme activity observed, but within 5 days of storing this fraction, it showed acid phosphatase activity. So, they thought that there must be somewhere that this enzyme had been sequestered, and after 5 days was released to show the acid phosphatase activity. So, de Duve again coined the term lysosome, and in 1963 coined the term autophagy, and also endocytosis was coined by Christian de Duve. So, this is an image of this is a transmission electron micrograph which is showing an autophagosome, this large body, and a lysosome which is fairly small. So, autophagosome merges with the lysosome, and you can see these lines which are the microtubules. Microtubules also play a role in movement of such autophagosomes towards the vacuoles. Also, they help in the movement of small endosomes towards the lysosome. Another Nobel Prize in the year 2016 has been to Yoshinori Oshumi, and it has created quite an interest in autophagy, in fasting, and in how regulation of calorie can really reverse aging. So, with this new glamorous role of autophagy, we will now talk about autophagy. So, it started off with in the year 1990s when Yoshinori Oshumi in Tokyo University decided to study autophagy. He had quite a simple plan, and he wanted to see if autophagy existed in a unicellular organism, and the organism of choice was yeast, and he developed several yeast strains that lacked the vacuolar proteases, proteinase A and proteinase B and carboxypeptidases. So, when such mutated strains were grown, it was found that autophagic bodies would accumulate in the vacuoles, and when such yeast were grown in nutrient deprived or in starvation conditions, they saw very large vacuoles which were even visible under light microscope. So, he wanted a low tech solution, he wanted a simple organism in which he could observe the formation of such large vacuoles. Later on, he went on to form several random mutations in the yeast which was already lacking the vacuolar proteases, and he was trying to see if there could be a mutant in which there is no vacuole formation at all. So, he was able to identify such a mutant which did not form any autophagic body, and he called this as the vacuole gene of autophagy 1, APG1, and after this he did several studies. About 75 mutants were made, and about 15 genes were found to be essential in autophagy, and these genes are now called ATG genes, and we will be discussing more about these genes in our coming lectures. Introduction to proteins, protein degradation and recycle in the lysosomal pathway. So, the Nobel Prize for the year 2016 in physiology or medicine was given to Yoshinoro Oshumi for his contribution or discovery of mechanism of autophagy. And this is the paper published in the year 1993 in the Phelps journal, which talked about isolation and characterization of autophagy defective mutants of Saccharomyces cerevisiae. So, Oshumi cloned several ATG genes and characterized their function. So, each gene was taken and its function was characterized. Apart from the ATG1 gene, which was a serine threonine kinase, others were very difficult because they were de novo genes and there was no existing information about their function which could be derived from their amino acid sequence or from the gene sequence. But because yeast genome was sequenced by the time this discovery was made, several or 15 such ATG genes were suggested to have a certain function, and a function was ascribed to them. So, it was found that the ATG1 protein forms a complex with the product of the ATG13 gene, and this interaction is regulated by the target of rapamycin kinase or TOR kinase. Here we are talking about TOR kinase because this is a selectivity checkpoint where nutritional status or ATP status of the cell, glucose status of the cell is monitored by the TOR, and it is very active in cells which are growing under nutrient rich conditions, and it hyperphosphorylates the ATG13 gene, which prevents the formation of a ATG13 and ATG1 complex. However, when there is starvation, when there is nutrient depletion, this TOR is inactivated, hence the ATG13 is dephosphorylated and it will bind to the ATG1 and the pathway which we will see now will be activated. So, a very small step which can be regulated by the nutritional status can regulate if autophagy is going to be switched on or switched off. So, this is the very exciting discovery which people now view from every viewpoint, how to deal with diseases, how to deal with cancer. So, what are the benefits of having a short term starvation when there is no malnutrition, when there are nutrients but there is low calorie. So, can organelle turnover be initiated, protection against metabolic stress, DNA damage species can be while there is DNA damage, several adducts are formed, those can be taken care of, can there be clearance of aggregation prone proteins, then regulation of the cell size. So, there is endosome formation which takes up the plasma membrane and engulfs it and sends it to the lysosome, regulation of the organelle size, longevity related to anti-aging, then programmed cell death, which is very important when we talk about tumors and cancers and finding cures for them, and defense against intracellular pathogens, because autophagy encounters or encompasses taking up large cellular components and degrading them. Also here autophagy is also responsible for forming certain molecules which have to be give immunogenicity. So, it is very important for MHC2 class of molecules where peptides are formed and are shown by the MHC2 to be recognized by the T helper cells, cytotoxic T helper cells. So, autophagy, what is autophagy? Autophagy means eating self, and here the eating self is the cell is eating its own components. So, there is rearrangement of subcellular membranes to sequester the cytosolic components and organelles and proteins and bring into the lysosomal degradation. Once degradation has occurred, there is removal or transporting out of the small components of or amino acids or DNA to the cytosol. So, there is recycling of the intracellular components, there is recycling of the intracellular organelles. Any organelles such as mitochondria, perioxosomes, Golgi, so they also encounter recycling. Then energy homeostasis when there is cellular stress. So, autophagy also when there is protein degradation and release of the amino acids etc., there is also release of energy which helps in fathoming the cellular stress of starvation. So, this autophagy is not a very simple process, it is very complex, highly coordinated and conserved across the yeast single cellular yeast to the mammals, and it is evolutionarily conserved. So, these autophagy related genes, the ATGs, and about 40 to 45 genes have been identified in yeast and mammals, and most of them you will find homologous functions, and they are responsible for removing the damaged cellular components such as mitochondria and long lived protein, and it is a catabolic mechanism. When we talk about long lived protein in ubiquitin proteasome pathway, we were talking about regulating the protein concentration and fine tuning it to the metabolic need of the cell. In that case, a newly formed protein, if requires a change in its concentration, then it was ubiquitinated and proteasome would degrade it to maintain its concentration. But in case of autophagy, most of the long lasted or the long lived proteins, damaged proteins which come in the category of the fibrils and the plaques that are formed in the neurodegenerative diseases can also be taken care or can be degraded by the lysosomal machinery. So, autophagy is of three types. The first one is the macroautophagy. In this there is the engulfment of cellular contents, organelles, large proteins which have been misfolded, and even invading microbes and viruses can be taken up by microautophagy. Here the AT gene 5 to 12 shows conjugation and it interacts with the ATG16, and this multimerizes at the phagophore and LC3 processing occurs which inserts this into the existing phagophore membrane. So, here it is forming a autophagosome. we will look into the process in detail. A phagophore is the beginning of the of autophagy. It is a small cup-like structure, and it eventually grows and forms the autophagosomes. The membrane which is forming the phagophore may come from the plasma membrane, it may come from the Golgi, or sometimes even from the ER. So, the autophagosome once formed, the phagophore is sealed off, it forms a fairly large vacuole which is 300 to 900 nanometer and hence visible under light microscope. It is the beginning of the endocytic pathway. So, then there is acquisition of acidic and degradative enzymes. The lysosomal proteases will now come from the Golgi and fuse with the vesicle with this autophagosome. Then there is trafficking along the microtubules, as we had seen in the transmission electron image, that microtubules are associated with the autophagosomes and they will push the autophagosome towards the proximity of the lysosome for further degradation, fusion and further degradation. Once degradation has occurred by hydrolysis, it will result in recycling of the nutrients, and now the lysosomal membrane permeases and transporters will export these nutrients out to the cytosol. This way membranes will also be recycled and energy is also released, and this will help in during this process only several components of the TCA cycle will be formed, and they can now become part of the TCA cycle and help in ATP generation. So, amino acids which are released in the muscles and liver after autophagy are used for gluconeogenesis in the liver to produce ATP by sending all these components into the tricarboxylic acid cycle. Degradation of liver lipid droplets by autophagy via lipophagy contributes to the generation of free fatty acids that are oxidized in the mitochondria, and under normal pathological processes there is increased lysosomal activity when there is muscle wastage, as in myopathies, during disuse or traumatic injury. Even after childbirth there is regression of the uterus by the autophagic process. Involution of mammary post the lactation also is by autophagic process. Chronic inflammation diseases such as rheumatoid arthritis, Crohn's disease, etc. Also are due to autophagic process or due to some disruption in the autophagic process. So, second type of autophagy is microautophagy. In this you do not encounter very large organelles or bacteria or viruses, but here there is cytosolic cargo which is engulfed by the lysosome directly by invagination, and in this the lysosomal membrane randomly invaginates and differentiates into an autophagic tubule to enclose the portion of the cytosol, and then the vesicle from the top of the tube will fuse and it will bud into the lumen within the lysosome. This is important for pruning the size of the organelles, in membrane homeostasis, in maintaining the composition of biological membranes, and survival of cell under nitrogen stress. Microautophagy is sensitive to GTP concentrations and it is modulated by calmodulin, and the non-exclusive microautophagy will engulf soluble intracellular substrates by tubular invagination in mammals. It is very frequently seen, while in yeast you may encounter selective microautophagy, which is micropexophagy, which is taking up of the perioxosomes, or piecemeal microautophagy of the nucleus, in which pieces or parts of the nucleuses after being tagged are taken up by the lysosome, and this will sequester specific parts or specific organelles with its arm-like protrusion, and this is found in yeast. So, disruption in micro-autophagy can result in several diseases, and neurodegeneration, Gerstmann-Straussler, Skinner disease, Huntington's disease, types of albinism, Chediak-Higashi syndrome, and glycogen storage diseases, which we discussed, Pompe's disease, can occur if there is any disruption in the micro-autophagy. The third type of autophagy is chaperone-mediated autophagy, in which there is a motif which is KFERQ containing protein containing these 5 amino acids is recognized by the molecular assistants or chaperones. Here it is called the heat shock protein HSC70 or the heat shock cognate of the HSP70 family, and this HSC70 acts as a mediator. It binds to the motif of the protein which has to be degraded and brings it to the LAMP2A, which is the lysosomal membrane transporter. So, this lysosome associated membrane protein LAMP type of 2A is the receptor for HSC70. This LAMP2A will internalize and will take up the translocation of the unfolded polypeptide into the lysosome. Once it is been internalized, it will undergo degradation by the acid hydrolysis. Lysosome will show uptake of several components, and here we are depicting about 14 types of lysomophagy can occur. So, it is of all types are summarized here, taking up of smaller organelles to larger mitochondria, and all have been summarized here. Give a look to this. So, when there are several type of organelles are taken up by the lysosome through phagosome formation, then you give them different names. If it is ribosomes, then ribophagy, mitophagy in case of mitochondria, xenophagy in case of viruses and bacteria, aggregophagy for taking up the protein aggregates, and lipophagy when taking up the lipid droplets. Very essential, lipid droplet uptake very essential in lipid metabolism in liver and glycogen storage. So, this can be both selective and non-selective mechanism. So, when we have discussed about several genes being responsible for autophagosome formation, let us briefly go through how these genes are regulating the formation of autolysosome. So, there are two complexes which are formed, which is the class 3 phosphoinositol 3 kinase complex. It is containing all these components ATG1, 14L, Beclin, Ambra, etc., and the ATG1 ULK1 complex, which is containing ULK1, ATG13, ATG10, FIP200. Other regulators are also present, ATG2A, B, WIPI1 and 2, DFCP, etc. So, all of these are going to cooperate in the autophagosome formation. There are two ubiquitin like mechanisms, as we had said earlier that ubiquitin like tagging also occurs in lysosome for selectivity. So, these two novel protein conjugation systems are very necessary for autophagosome formation, and they are the ATG12 to ATG5 and ATG8 phosphatidylethynolamine systems. So, they have the components similar to ubiquitin E3 in ligases present, and the two mechanisms are ATG5 and ATG12, which are conjugated and they bind to ATG16. Secondly, there is a pre-LC3 which is cleaved by the protease ATG4 and binds to the lipid phosphatidylethanolamine PE which facilitates its anchoring to the autophagosomal membrane. So, once formed, the autophagosome then fuses with lysosome or endosomes, and this process will again encounter several lysosomal proteins such as LAMP and RAB, and after degradation of the lysosomal of the autosomal components, there is release via the permeases, and a permease enzyme or permease gene is the ATG22 in the yeast, which is present in the lysosomal membrane, and then there is all these are translocated and translocated to the cytosol, where they can partake into new anabolic reactions. So, there is recruitment, and then there is energy sensitive modulation by TOR kinases also going to occur in this, and this will result in formation of an autophagosome, then fusion with the lysosome. As we were talking about CFTR, which was responsible for acidification, also there are other components which are responsible for fusion of an autophagosome to a lysosome. So, this diagram briefly is telling you that there is first formation of a phagophore which forms a autophagosome, which eventually fuses onto with the lysosome with the help of SNARES and microtubules and other components, and will result in an autolysosome formation where actual degradation occurs. In this component only there is release of the building blocks, amino acids, lipids, etc. for further via the permeases for further anabolic reactions. So, what is the nutritional signaling, or what kind of signaling can occur in a autophagous pathway? So, there is upstream signaling of the ATG1 complex which regulates the autophagy. So, ATG1 is the initiation step for autophagy. Growth factors are important, energy status, the amino acids, insulin, ATP levels, glucose, glucagon, all are responsible for modulating the regulation or regulating the autophagy. mTOR C1 kinase integrates the nutritional cues which it is getting from the environment and surrounding, and it will actively suppress the macroautophagy when there is normal nutritional condition, when the cell is flourishing. But when there is starvation or nutritional stress, or there is nitrogen stress, that means there is no nitrogen or amino acid in this media, then mTOR C will be inhibited and activation of autophagy will occur. Also, there is homeostasis of energetic balance that can be maintained by autophagy, thereby recycling of the amino acids to sustain protein synthesis of essential proteins. Come to think of it, when a cell is under lot of stress, most of its luxurious proteins will be taken up by the autophagosome and merge with the lysosome to release the amino acids, so that only those very essential proteins can be formed which are important for housekeeping function, and most of the luxurious or other activities are stopped. So, this way recycling is occurring and energetic balance is maintained even in the absence of amino acids from the external media or surrounding. Glucagon also exerts stimulatory effect over macroautophagy, and insulin is an inhibitor of autophagy. Amino acids can be directly utilized to obtain ATP through their entry into the Krebs cycle, and hence again you have generation of energy to fathom the stress conditions. And we were discussing about role of microtubules and SNARES in fusion of autophagosome with the lysosome to form a autophagolysosome. So, the soluble N-ethyl methylamide sensitive factor, attachment protein receptor, SNARE, are basic elements which are involved in intracellular membrane fusion, and they assist the complete fusion between the autophagosome and the lysosome, and the microtubules are essential for movement or centripetal movement of the autophagosome towards the lysosome. So, they nudge the autophagosome towards the lysosome for autophagy. Selective autophagy. So, autophagy is a very selected process, a highly regulated process. In this case, if a certain cargo or certain protein is selectively tagged, then these cargo receptor proteins tether to a cargo to a nascent or a newly formed autophagosomal body by binding to the cargo and the ATG8 and LC3 family of proteins on the isolation membrane. So, several receptor proteins recognize polyubiquitin chains attached to the surface of the cargo for selective autophagy. We have studied about polyubiquitin tagging, which results in tagging it towards the proteasome, but here we will encounter how some receptors which are important in autophagy also require a polyubiquitin chain. Sometimes even a single molecule of ubiquitin attached to a protein is sufficient for selective autophagy. So, there is selective rerouting of the selective pathway proteins destined for the plasma membrane, and they go on to transfer to the Golgi and the trans-Golgi network. So, proteins to be degraded are polyubiquitinated and are sorted to the lysosome, and here ubiquitin is signaling for a lysosomal degradation and not a proteasomal degradation. So, this is seen in yeast cells which are growing in nitrogen rich media, and the demand for uptake of amino acid surrounding is reduced because there is plenty of it there. So, permeases which are transporters are not required anymore, and hence they have to be degraded. So, they are quickly ubiquitinated and degraded by the lysosomal pathway. So, autophagy is very selectively induced by starvation as to maintain the energy homeostasis and recycling and degradation of proteins and lipids and DNA, and it is also encountered in developmental and differentiation stages of the embryo to eliminate the maternal mRNAs and paternal mitochondria. Even when the placenta has ceased to provide nutrition, it helps in maintaining the energy balance. Differentiation of erythrocytes, lymphocytes and adipocytes is occurring by autophagy. Homeostasis of immune cells and regulation of self and non-self. So, there is self tolerance, which is again by autophagy. Induction of autophagy during calorie restriction, and then there are anti-aging effects which may occur because of autophagy which can be induced by starvation. So, I will just show you one. So, this is the recent NCBI website, we are looking at PubMed. So, there are several research which is going on relating fasting to autophagy and what are the benefits and how disease can be modulated. In our future lectures, we will be discussing about autophagy related disease and how it is important for cancers, neurodegenerative diseases, metabolic diseases such as insulin and type 2 diabetes, and how autophagy can modulate aging processes. So, we will briefly pause in this lecture.
With this note, we would like to thank Dr. Unnati Gulati for giving us a very productive session. Dear friends, if you want to give your feedback for this particular lecture, then you can mail us at info.cc@nic.in. We would be meeting again soon. Till then, take care. Goodbye. Thank you, ma'am. Thank you so very much.
Thank you, Kitika.